Welcome to San Francisco, and welcome to the future, because you are the future in this room. And we're so proud to have you here. I want to make sure that I acknowledge some of our board members who are here. Dr. Francisco Prieto, doctor from Sacramento, expertise in diabetes. Dr. Bert Lubin, I think, is here as well. Alan mentioned him. There he is with the injured hand. We hope you get well soon, but thank you for being here. And of course to Dr. Townsend, who has worked with us so well in the last few years here as president of our institute. And of course, our thanks to Dr. Zambrano, Dr. Yaffe, and of course, she should receive an honorary doctorate, Maybelle Cortez, for all the work that she does here, and the rest of the CIRM staff that is here as well. Would all the CIRM staff please rise so everyone knows who you are? These are all our scientists here. As Alan said, don't be afraid to ask them questions. They ask a lot of questions of us all the time, and I think it's important for that interaction to take place. The Bridges program is probably one of the programs that the legislature and the members of Congress know a lot about. As a matter of fact, last week I sent a personal letter to each member of the legislature in California and each member of Congress to let them know in their districts what Bridges programs were being funded, so that they know that you're working in their area, so they're aware of what you're doing and they're aware of the promise that you hold for all of us. We recently, as a board, approved $27 million more to make sure that this program continues for another 3 years. And as Dr. Zambrano said, if it wasn't for the leadership of Dr. Townsend and, quite frankly, Dr. Olson, who was very, very supportive, as well as Dr. Yaffe and Dr. Zambrano, making sure that this program remained within the embrace of CIRM, because we recognize how important it is for the future. We also want to make sure that you receive a special acknowledgement and welcome, although he's not here today, from our new chair, Jonathan Thomas, who's from Los Angeles but could not be here today, but also to make sure that you realize that we are really, at the end of the day, a California state agency. The voters created us as a state agency in 2004, Proposition 71. We are a state agency which is also unique. We operate with a 6% cap on operating costs, which means 94% of all the funds that we raise through the sale of general obligation bonds goes directly to research. In addition, you should know that between now and 2014, this agency will generate over $201 million in new revenue for the taxpayers of California. I don't think you can say that about the DMV or any other state agency. And I think it's important for you to carry that message across so that people realize that this is not just a funding agency, but quite frankly, this agency has not cost the taxpayers of California 1 cent yet, because of the incredible opportunities we've had to capitalize the bonds and to extract new revenues for the state of California. And I'm sure you'll hear about it later as to what treatments emerge from this research that will reduce the cost of healthcare and agony and pain in California and, quite frankly, in the world. As a colon cancer survivor, I'm proud to be part of CIRM. But more than that, I'm proud of you, because you really represent the future in this state. And I just wanted to make sure that you understood how important you are to the members of our board, obviously to our president, to the staff here at CIRM, and make sure that you utilize the time that you're here to the best advantage you can to exercise your ability to ask those questions, to exercise your ability to have dialogue with each other, but also with the staff that's here and the wonderful presentation that you're going to have, I know, from our wonderful speaker tonight, Dr. Lebkowski. Jane, pleasure to see you here. Take care and good luck. Dr. Pat Olson.
Good evening. It is my very great pleasure tonight to introduce our speaker, Dr. Jane Lebkowski from Geron Corporation. It is fitting that as CIRM expands its funding program into therapy development, that we have as our keynote speaker tonight, Dr. Lebkowski, who has pioneered the development of cell therapies derived from embryonic stem cells. Jane has been involved in novel therapy development actually for most of her career. She was initially Vice President of Research and Development at Applied Immune Sciences, whose focus was on innovative cell and gene therapies, products designed to enhance and restore immune function. Following the acquisition of Applied Immune Sciences by Rohn Planck Rohr in 1995, Dr. Lebkowski was appointed Vice President of Discovery Research at that institution and had responsibility for preclinical research of gene therapy approaches for the treatment of cancer, cardiovascular disease, and nervous system disorders. You know, wanting an additional challenge above that of gene therapy, Jane actually left Rohn Planck Rohr in 1998 to join Geron Corporation and to be intimately involved in their embryonic stem cell program. She is currently Senior Vice President and Chief Scientific Officer at Geron. She heads their human embryonic stem cell program and is responsible for all research, preclinical development, product development, manufacturing, and clinical development for that program. Under her leadership, Geron filed the first IND and received the first approval from the FDA to initiate clinical trials for a human embryonic stem cell-derived product. It is this pioneering work that she will be speaking about tonight. Please join me in welcoming Dr. Jane Lebkowski.
Thank you. Sorry that I'm not going to be standing at the podium. I'm recovering from a hip injury and I wish that I'd also started some projects involved in orthopedics. But what I really want to talk about today is some of the work that we've been doing at Geron over the last 10 to 12 years to really develop a human embryonic stem cell derived oligodendrocyte progenitor for the treatment of spinal cord injury and other CNS disorders. So when you think about developing a stem cell-based therapy, there are lots of things you need to consider. First, what's the clinical indication that you're going to use? Because the clinical indication will dictate what type of cell you're going to be looking at transplanting, what state of differentiation is going to be important, and how and when do you have to administer that particular cell. Again, you need to consider the source of those particular stem cells, whether they're allogeneic or autologous. You need to look at the desired differentiation status of those cells. And what is the formulation of those cells? Can you get enough cells into the area of interest so that they can be effective? What's the delivery route? Do you have to implant it through a surgical procedure, or can you give it systemically? How long do the cells need to survive? In some cases, for some types of cells, you might not need to have long-term survival. For other applications, more regenerative applications, you're probably considering long-term survival. Do you need immunosuppression? What's the mechanism of action that's anticipated? What are the potential benefits? Are there risks of infection as a result of the cells themselves or the implantation procedure? Are there various toxicological concerns? And what are the tumorigenicity concerns? Not only tumors or malignant tumors, but any kinds of tissue that might not be wanted in the place where you're injecting the cells. All of these questions, plus more, really impact the design of the program that you're going to use to develop that particular therapeutic. I'm going to be talking about something called Geron OPC1. Geron OPC1, they're oligodendrocyte progenitor cells. They're cryopreserved and allogeneic cell population that are derived from human embryonic stem cells. They have a very characterized composition of cells. They contain oligodendrocyte progenitor cells. They produce neurotrophic factors, and they also produce induced myelination of denuded axons, and I'll be showing you some data on that. But the really intended application right from the start for this particular product, for this particular cell therapy, was to be used as an off-the-shelf product. We wanted to use it, have it available on demand for spinal cord injury and the treatment of other central nervous system disorders. So that's the fundamental of the cell therapy that I'm going to be talking about. What was the rationale for that? The rationale for these oligodendrocyte progenitor cells really came from looking at the pathology of the spinal cord injury site. So most spinal cord injuries are contusive in humans. And what you see mostly in most cases of spinal cord injury is that in the core of the injury, there's a zone of the original damage. And that through the influx of a variety of different excitatory factors, various neurotrophic factors, or the also inflammatory factors, that you can see that this zone of original damage usually enlarges to form a large cavity. And if you look very, very closely at the axons, what you can find is that there are actually three pathologies. What you see in some cases at the perimeter of the spinal cord injury is that you have an intact axon that has intact myelin. You will see in many other cases that there is in fact severed axons, so that there's no continuity or conduction down that particular axon. And then also the other pathology you will see is that relatively intact axons, but that they have lost their myelination, so that there is abrupt or there is discontinuous conduction down that particular axon. All three of those pathologies obviously have consequences. But the hypothesis is that if you could transplant oligodendrocyte progenitors into that spinal cord injury at a time point after injury, that these oligodendrocytes might be able to do a couple of things. First, oligodendrocytes in their progenitors produce neurotrophic factors, and those neurotrophic factors might aid in the repair or actually sparing of tissue at that particular site. The second thing is that these oligodendrocyte progenitors are actually the precursors of oligodendrocytes, and it's oligodendrocytes that are important in remyelinating axons that are denuded. So together with Hans Kierstedt at the University of California in Irvine, we developed a process to differentiate human embryonic stem cells into oligodendrocyte progenitors. And that work has been published, but essentially what we do is make banks of human embryonic stem cells, which are the starting material for the process. We then take vials of these banks and expand them. We then subsequently take these expanded cells, differentiate them into the oligodendrocyte progenitors, and then they're harvested and cryopreserved for later use. And these are just pictures of the cells at the various stages with the final product at the end. It's important to know that whenever you're differentiating an embryonic stem cell, or any stem cell for that matter, that the differentiation is not synchronous, it is not, in this case, uniform. So that what you have is a mixed population of cells at the end of your differentiation process. So that what we do is you take undifferentiated cells, and using a variety of different factors, we preferentially drive it down the differentiation path through the neural lineage. And then you, depending on the combination and the timing of growth factors, you can get neural stem cells, oligodendrocyte progenitor cells, and if you carry it on longer, even looking at more mature neurons, astrocytes and oligodendrocytes. The population that we're using, Geron OPC1, it basically is a population of cells that has a distribution around the oligodendrocyte progenitor population. And we use markers for undifferentiated cells in all the different stages of differentiation to try to characterize that population, to understand what is the predominant stage of differentiation. Are they mature? Are they immature? And do they have, in particular, undifferentiated cells as part of that particular population? In characterizing these cells, it's also important to look at the populations that might have not gone down the neural lineage. For instance, evidence of mesoderm, endoderm, or in fact, one specific example, epithelial cells. So it's really very important to characterize the stage, the content, the composition of the cells that you're looking at, because that will affect the safety and efficacy of your particular cell therapy. I cannot emphasize more through direct experience in developing this particular cell therapy that keeping cells from every animal experiment, every in vitro experiment that you have, keep retains of those populations to do thorough calculation of your populations. In our particular sense, we looked at whole genome microarray analysis, quantitative RT-PCR, flow cytometry, high content imaging, and biological assays to really characterize each production lot that you're making and looking for subtle differences in those particular populations, because it really can affect what you see at the end in your animal studies and in humans. It's really important to correlate this composition with the in vivo activity of these cells in animal models, and it's really very, very, very critical for establishing your cell composition specifications, because you have to have various specifications for your product before you can release it for the clinic. And that leads me to what are the kinds of things that you have to know about your cell therapy before you can release it for human use. In this case, after manufacturing, we look at the morphology, the viability, the cell composition, cells that are having an oligodendrocyte progenitor phenotype. We look for various extraneous phenotypes, and extraneous phenotypes is just a terminology that the FDA uses for cells that you might not want in your product, whether that might be harmful or not harmful, but just there. And in particular, for an embryonic stem cell-based therapy, you really want to look for undifferentiated cells, because these could be the source of teratoma formation. Obviously, also need to characterize your cells for adventitious agent testing, sterility and mycoplasma, and also endotoxin testing. So again, very important to characterize your cell population, knowing what cells are in there, knowing what cells might potentially cause problems, what cells are causing your efficacy. We know that OPC1 can mature into oligodendrocytes. This is OPC1 as it's thawed, but we can mature it in vitro to form these nice oligodendrocytes. We know that it's important also to characterize the function, not just a phenotypic marker, but the actual function of your cells. In this particular case, we know that OPC1 produces neurotrophic factors. For instance, if you take, in this case, rat sensory neurons, and you expose them to conditioned media from the OPCs, what you find is that they produce and extend these neurites, suggestive of the projection of neurotrophic factors. And we've gone on to characterize what neurotrophic factors are produced, at least in physiological concentrations in vitro. And there's a whole series of them that are produced. We also know that these cells can produce myelin, that they can wrap denuded axons. So for instance, if you take these OPCs and you transplant them into a mouse model, in this case a shiverer model, in this case that is defective in myelin basic protein and is a dysmyelinated phenotype, as shown here. If you the animals that are transplanted with the OPCs show the production of these thick myelin sheaths that are produced, that wrap the axons in the area where there are cells. We also have some data now suggesting also that they can function to myelinate in monkey models. So it's important, again, to show that these cells have the potential, at least, for producing myelin. We also know that these cells can induce neovascularization. So in the areas around the grafts what you can see are the presence of these vessels of host origin that are surrounded by the OPCs. And so in animals that are treated with OPCs, we can see around, particularly surrounding the lesion site, evidence of neovascularization. Again, suggestive that these cells might have induced vascularization and have some reparative effect. So to try to characterize the function, or the more therapeutic function, the clinical function of these cells, we've used a variety of different injury models. There predominantly been rats, and rats that have a thoracic injury, that is either a bilateral contusion model in the thoracic spinal cord at T10, or a cervical unilateral injury model, or a bilateral injury model at C5. In general, what we do is we transplant at 7 days post-injury, and the cells are injected right into the spinal cord injury site. And they're generally transplanted somewhere between 7 and 10 days post-injury. And the reason we do it at that period of time, the subacute injury period of time, is that data has shown us that if we transplant post 2 months injury, we don't see any therapeutic effects. We only see it when we are injecting in, so far, the 7 to 14 day window. We're looking at some intermediate time points right now between 14 days and 2 months. But it appears at least that these cells are effective at 7 days post-transplant. And what we do is we evaluate the animals. We look for clinical efficacy, histological efficacy, looking for cell survival, phenotype, migration, and toxicity. And to make a long story short, this is some work that was published by Hans Kiersted and also some work done in our laboratory to show that when you transplant Geron OPC1, these oligodendrocyte progenitors, into either spinal cord injured, thoracic spinal cord injured rats, or in cervical injured animals, we can't see improvements in their locomotor activity. So for instance, in the thoracic injury model, I won't go through all of the data right now, but we can see that there is an increased weight-bearing activity of the animals, improved hind limb, forelimb coordination, improved hind paw clearance, improved trunk stability, and in decreased tail drag. Again, all indications of improved motor control in these animals. They also have increased stride length, decreased stride width, again, consistent with the increased motor control in these particular animals that have received these oligodendrocyte progenitors. Importantly, also, we find histologically that there seems to be a correlate between the improvement in the locomotor scores and also what we see histologically in vivo. So for instance, if we look at, in this case, in a rat that has 9 months after injury and transplant, in this case with vehicle saline, what you find, again, as I showed you in that one diagram, is that you have a very large cavity in the center of the spinal cord at the injury site. And if you look at a high power magnification, what you see is that there's this large cavity that is surrounded by a glial scar, and repair through that glial scar is particularly difficult and doesn't occur. However, if you look at an animal that was transplanted 7 days post-injury, and you look at that animal 9 months post-transplant, you can see that there is considerably less cavitation in the spinal cord, and in brown, as detected in this case by in situ hybridization for a human ALU sequence, you can see that there are lots of, in brown, human cells that are surviving in that particular spinal cord. And also, more importantly, if you look at the center of this particular lesion as expanded here in this particular photograph, you see in blue that there are these fibers that are traversing the injury site. And the blue myelin stain, Luxol Fast blue stain, actually indicates that there are surviving and myelinated fibers, nerve fibers, that are actually traversing the injury site. Very, very, very exciting results. Again, suggestive that at least either through a tissue sparing effect or through myelination or through these neurotrophic factors, that we are seeing some preservation of the actual structure of the spinal cord. We can characterize what kinds of cells are present in the spinal cord, and we've done that pretty extensively right now. We see that the cells are well distributed throughout the spinal cord, that the predominant phenotype are oligodendric glial cells or other neural cell types. They do mature to form mature oligodendrocytes. They do this very slowly. It takes weeks and months. After about 8 weeks, you see evidence maybe 5 to 10% of the cells are oligodendrocytes. But it's really this slow maturation over approximately 26 and greater weeks that we see more and more presence of mature oligodendrocytes. We see obviously no evidence for uncontrolled cell growth, which is an important safety factor. So it's really those studies that are the proof of concept that these cells could have potential therapeutic application. And then through a series of various biodistribution studies, dosing delivery studies, toxicology studies, tumorigenicity studies, and immune rejection studies that would actually look to verify, in fact, that these cells have efficacy, but also have a safety profile that justifies their use in humans. For all of the studies that I'm going to be talking about, you have to select a dose that is representative of the dose that you're going to put in humans. For our particular biodistribution and safety studies, we used 2 different doses, which one was 2.4 times 10 to the 5th cells, which was the lowest efficacious dose in a rat. And based on the actual architecture and geometry and volume of the spinal cord, we conservatively selected a dose of 2.4 times 10 to the 6th cells as the human equivalent dose. And those 2 doses were used for all of the studies. An important feature to look at is what is the distribution of these cells when you're injecting them right in the spinal cord. Do they stay in the spinal cord, or do they distribute outside of the spinal cord, especially because of the injury? We looked at a variety of different studies using quantitative PCR to track human cells in the spinal cord, the brain, and a variety of different other sites outside of the central nervous system. And again, to make a long story short, we found that most of the cells are found in the epicenter of the lesion, that if you look 2 centimeters rostral of the lesion, you can see that there are still some cells, and that at 4 centimeters, you can see a diminishing numbers of cells. So there's a gradient of cells. Similarly, we found that there was no evidence of cells outside of the central nervous system. So that was good. The idea here was that even though there is an injury there, the cells using the methodology that we're using in the rats, which mimics that which we're going to use in the clinic, that the cells primarily essentially stay within the central nervous system. And they don't migrate for huge differences. You don't see them, for instance, when you inject in the thoracic cord, you don't see them in the brain. We looked at toxicology studies. We looked at 2 doses in rats for a variety, for 2, 6, and 9 months post-transplant, and looked at a variety of different parameters, the toxicity of delivery, the mortality, adverse clinical observations, systemic toxicity, hematologic toxicity, coagulation problems. We've looked for clinical chemistry, macropathology, micropathology, and also, importantly, allodynia. Because in patients with spinal cord injury, the last thing you want to do is to induce potential neuropathic pain that could be very, very debilitating for individuals. Based on these results, we saw no evidence of adverse clinical effects. The big question was, do you see teratomas? And the answer is, so far, with materials that pass our clinical specifications, we have not seen evidence of teratomas in these animals even when we've looked up to 12 months with doses that are the same as our lowest dose that we anticipate in human clinical studies. We've looked now at over 1,000 animals that have received this material. So all of the tissues that were examined by a blinded independent veterinary pathologist. They looked at the spinal cord, they looked at through the cervical cord, they looked at 5 different levels of the brain, they looked at all peripheral tissues to look for the evidence of any teratomas, or for that matter, any kinds of masses, and did not find them. The only issue that we did find in some animals was the presence of these, what we would call epithelial cysts, that were microscopic in size. Some of them, the largest one was approximately 1 millimeter in diameter. These were found in some animals. We have now developed markers to really be able to screen our lots to make sure to minimize the presence of these kinds of epithelial cysts as in future lots that are going into the clinic. The other thing too is that we performed various spiking studies where we deliberately took undifferentiated cells and we spiked it into our product to see what level of contamination that we would need to see in order to get a teratoma. Again, to make a long story short, what we found was that if we saw anywhere from, first, if we looked at the product itself, we saw no evidence of teratomas. If we looked at animals that were injected with our product that was deliberately spiked with up to 5% undifferentiated cells, we still saw no evidence of teratomas. But if we had 10% or greater undifferentiated cells, we did see evidence of teratomas. So again, suggestive that you need to control the level of undifferentiated cells in your product so you don't see teratomas. And our specifications for undifferentiated cells are orders of magnitude lower than 5% undifferentiated cells. Finally, the last thing we wanted to look at was, what is the immunogenicity of these cells? We're going to be looking in the allogeneic setting. These are allogeneic cells that are going into patients with spinal cord injuries. Are these cells going to be rejected? Do we need lifelong immunosuppression? This is a very, very difficult question to answer in animal models. Most of the animal models to look for allogeneic effects for human cells are extremely difficult and challenging. One of the things we have classified and determined on these cells is we looked at that they do express class 1 on the surface of the cells. They don't express class 2. They don't seem to elicit reactivity from alloantibodies. They're not killed by human NK cells, and that they have very low T-cell immunostimulatory activities. So that where we're going now in the clinic is to look at using a low dose and transient immunosuppression during the initial period of time after injury, and once the inflammatory responses are dying down, we are removing that immunosuppression. So we've looked at a variety of different protocols. We've looked at a variety of different toxicology studies to establish the safety profile of Geron OPC1. And we filed a couple of years ago now our IND with the FDA to establish the first clinical trial with OPC1, which is ongoing right now. It's an open label trial. OPC1 is injected, used in patients with subacute, functionally complete thoracic injuries at levels T3 to T10. We're injecting a relatively low dose of cells right now, the equivalent of the lowest dose, which was active in rats. We're transplanting 7 to 14 days post-injury. Again, as I said, temporary immunosuppression. And the primary endpoint in this particular study is looking at safety. Overall safety, are there any problems associated with the injection of the cells, any problems with infection, any problems with the administration of the immunosuppressive agents. And then neurological safety. Is there any reason why these cells are, for any particular instance, causing any further neurological damage? Obviously, we're looking also for any evidence of masses or tumors or cysts that get developed. The secondary endpoints are activity. We're looking at the ASIA grade and score. ASIA is the American Spinal Injury Association grading system for determining the level of neurologic damage that is in patients. We're looking at something called the UAB-IMR, which is an indication of motor activity in the legs. Looking at independence measurements, basically quality of life measurements. Looking at bowel and bladder function, and also pain assessments. We're delivering these cells in the same manner that we did in the animals, in our rat studies. And to do that, we actually developed this device, which is, we call this syringe positioning device, that actually assists the neurosurgeon in injecting the cells into the spinal cord. So again, these cells are injected in a separate procedure that is a separate neurological surgical procedure to implant these particular cells. They're injected in a 50 microliter injection, 5 millimeters caudal to the epicenter of the injury, and using this particular device, it can aid the neurosurgeons in actually the slow delivery of these cells, because you don't want a lot of efflux in the cells floating away. In the particular study schema, what you see is that patients initially have their injury, and they come into the site, the clinical trial sites, where they're given an MRI, a baseline MRI, to determine the level and the severity of their injury. If they pass a screening criteria, they are given an injection of Geron OPC1, in this case, again, 2 million cells, and then they're followed for the course of a year. And during that course of the year, they receive an additional 8 MRIs to look for any possible damage that might be associated with the administration of these cells. In addition, when patients sign on for this particular clinical study, they also sign on for a long-term follow-up study where they will be followed for an additional 14 years to monitor for any improvements, to monitor for any side effects or safety issues that might be associated with the administration of these cells. So just give you a update on the clinical trial and the first 2 patients in the clinical trial. Both of these patients have had successful surgery and no severe adverse events associated to date. Both patients, in fact, had a single neurological level of T6, so a thoracic level 6. There are, again, no SAEs to date. And in fact, the first patient now has completed a 180-day visit. There's no evidence of any change in neurological status at this particular point, and no expected adverse events, which is very, very good. One of the important things that we see in this particular patient is that this patient has not yet developed any evidence of cavitation, which is a good sign as far as we're concerned in terms of looking at the potential survival of the graft itself and potentially the preservation of tissue in the spinal cord injury site. Too early to tell, but encouraging sign. And then the last thing I want to say is that in our studies from the first patient so far, we see no evidence that there are developments of immunological rejection of OPC1. So where do we go from here? Our objectives really are to look at continuing the particular clinical trial in patients with complete thoracic injuries and given a low dose. Given safety in this particular cohort of individuals, we will be looking at expanding the criteria for the trial, looking at doing dose escalation, looking at including patients with cervical injuries, and then eventually patients with incomplete injuries, where there's likely a better potential for seeing efficacy in these particular trials. So you start off the trials in patients who have the very worst of injuries, and then you start looking at less severe patients where you have the potential also of seeing improved efficacy. We're also looking now at using these cells for other indications, because I told you that they produce neurotrophic factors, they can remyelinate axons, they can vascularize. So that's suggestive of tissue repair. And we're looking at trying to see if these cells will actually have effects, positive effects, in other indications. So we have some collaborations ongoing with Jeff Kosis to look for evidence in multiple sclerosis lesions. We're looking with Frank Laferla in an Alzheimer's disease models to see if these cells will have reparative effects. Paula Leone looking in Canavan's disease, a childhood adrenoleukodystrophy, again, where there's a deficiency in oligodendrocytes. And then finally with Michael Levy at Johns Hopkins to look at transverse myelitis. So again, looking at starting these applications and starting our studies in spinal cord injury, but using this potential safety and efficacy measurements that we can see there, plus additional preclinical studies to expand the applications of these kinds of cells to many neurological diseases. So I just want to summarize here and finish up by saying when you're looking at developing a program to develop a therapeutic cell type, we really need to think ahead to the end game and really very carefully design and execute your non-clinical and clinical studies. And you need to look at monitoring the safety and the potential efficacy of these studies. And that there's really risks that you need to take into consideration and developing your non-clinical studies to really test those particular risks, looking at the survival, rejection, integration, proliferation, and ectopic tissue formed by these cells. And that last thing, when you're looking at translating into the clinic, looking at trying to develop strategies so that if there are adverse effects, that you can detect them as quickly as possible. It's a really exciting area. It's a really exciting, a lot of potential for all of these stem cell products. And it's very, 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 very important that we do the diligence and the appropriate studies to actually accelerate the development of these kinds of therapies. So I'd like to end by thanking the team at Geron who's been involved in developing OPC1, the many committees, our data monitoring committee and steering committees, who have had a lot of input into the design of the clinical trial and in the non-clinical studies that were going to support the trial. Our radiology and clinical outcomes committees, who are important for helping us monitor and see that what effects we're having on our patients. Obviously, our clinical investigators at the 7 sites throughout the US that are executing this trial right now, and Hans Kierstead and his laboratory for the early work that they did in this program. Thank you.
Okay, so if anybody has questions, feel free. We have 2 microphones in the alleyway, so please come up and ask any question you'd like. Remember, no question is silly.
Yeah, that's a good question. We are not anticipating using a repeat dose just because this has to be implanted in a surgical procedure. The second thing is that the data that we have suggests that if we do the first dose at a time point after 2 months, that we don't see any efficacy. So we don't have any data to suggest that a repeat dose would be advantageous. And secondly, we probably wouldn't put patient through a second surgery.
I was curious how these cells are actually getting to the patients. Are they coming straight out of cryo, or are they in culture for a period of time? And then a follow-up to that, what steps need to happen within the medical community to prepare neurosurgeons for the sensitivity of cells coming out of cryo or cell culture to make regenerative medicine a reality?
Excellent question, okay? These cells are cryopreserved, they're thawed, they're washed at the site, and they're injected. There is no culture procedure at the clinical trial site. So they basically, in most of the cell processing laboratories at the site, what they do is they thaw it, concentrate it, wash it twice, and formulate it in Hepes buffer saline for injection. What is done to prepare the neurosurgeons for, in this case, the injection of these cells? That's a great question. We have a training procedure right now. Every neurosurgeon that is in our trial goes through a training procedure on a cadaver on the injection of these particular cells. So no one is allowed to inject in a human until they have been trained specifically by the neurosurgeons who developed this procedure, Dr. Rick Fessler at the University of Northwestern University. And they go through that extreme training procedure. So, and prior to clearing a site, they go through a dry run. Okay, so the dry run is at the site. All of the people involved in either prepping the cells or injecting the cells, they're in the OR. They're assembling this the syringe positioning device, and they are injecting the cells into a ball of cotton. So yes, there's a lot of training that goes on. Yeah.
Yes, two questions if I can. One is how do you assess for allodynia in the rat? And the second is why the site of injection 5 centimeters caudal to the site of injury was chosen?
That's two good questions. Allodynia, we measure allodynia in the rat in terms of sensitivity to hot and cold. We also look at mechanical stimulation of the paws using probes. So those are the methodologies that we have used, various light touches to see if there are any noxious responses to normally non-noxious stimuli. We also look for any evidence of self-mutilation, okay? And don't see that. But the primary is looking for sensitivity to light touch, pinprick, and hot and cold. The second question was, why 5 millimeters? It's 5 millimeters caudal of the injection site. That is something that for the lowest dose, we are going to be injecting into the spinal cord injury site, and the FDA asked us to go caudal, okay? So not for any particular reason, but more concern of, again, a little safety measure. So it's 5 millimeters caudal, and that was based on an FDA request.
Okay, Kelly.
Hi. So I have a question about the cell product. So you mentioned that it's actually a mixture of cells, and I wonder when you prepare your lots, how much variability is acceptable, and what is the purity of the OPCs in that lot, if you know it, and how important do you think actually having a mixture of cells as opposed to a single purified population will impact on the functionality that you might expect to see in the future?
Yeah, we have tight ranges on the various specifications for the cells, okay? And look for, again, the presence of undifferentiated cells, neural stem cells. We look for oligodendrocytes. We look for astrocytes. We look for neurons. So there is some leeway, okay? But basically, what you have to show is that you can repeatedly produce your cell product. What was your second question? It was the...
Whether or not you think that having a mixture of
Oh,
cells will be
yeah.
important.
You know, one of the things that I can't answer that question here. My bias is that having a mixture of cells is actually important, okay? We can't tease apart the different functions of the different cells that are in there. But my bias also is that you probably can't achieve a homogeneous purified population of cells because you'd have one cell. I mean, if you look at quantifying these cells and trying to identify the composition of cells, you see that they're at all different states, you know? So you can start subfractionating and subfractionating and subfractionating cells. And I think that it would be extremely difficult to say that you have a homogeneous population of cells. It's you know, like for instance, we're working also on product for cardiomyocytes, and we can say, wow, we've got a population that is, you know, greater than 70% alpha-actinin cells. But there's still, if you look at those cardiomyocytes that express alpha-actinin, they are still heterogeneous, you know? So it's a tricky business to try to, you know, say, I'm gonna get this purified population of cell. Yeah.
Hey, so while you have a heterogeneous mix, you don't have any MHC class 2 being expressed on it, but you do have MHC class 1. So do you think it's just because the graft is sequestered initially that you don't get an immune?
You know, it's really interesting in terms of the MHC class 1. I mean, we know from characterization, we're not seeing any hematopoietic cells or immune cells. We're not seeing T cells or anything like that in this particular population. We have deliberately tried to expose them to things like gamma interferon and other inflammatory factors to see if they would upregulate class 2, and we don't see that. Not to say that it won't happen in vivo, okay, but we haven't been able to achieve that yet. So I don't have the answer to why we don't see class 2 expression, especially when exposed to a variety of inflammatory factors, but we don't see it.
Well, I guess the mechanism for which you don't, while you have MHC class 1, you're
Right.
not getting an immune response.
Oh, so the
So you're
question is,
getting MHC
since we have
class
class 1, why aren't
Yeah.
we seeing?
Yeah, things being expressed through class 1.
Yeah, it's, you know, I don't know. We are not seeing very high levels of, we're not seeing very high levels of allostimulatory activity. My guess is that these cells are producing something that's inhibitory to an immune, to proliferating T cells. It has been described in the literature, for instance, that mesenchymal stem cells produce inhibitors of T cell responses. It's certainly been described by various laboratories that neural progenitors have the same effect. We, if anything, see a similar effect, but again, haven't characterized what that factor is.
And then just a real quick question. You pursued the rodent model. Did you ever go after non-human primates to do any of your testing?
The answer is we're looking at some non-human primates now for MS models, but the answer was no. The number, in terms of, it depends on what you want to answer, the question you want to answer in the primate studies. And in this particular case, looking for efficacy in the primate models, there just aren't any good primate models for efficacy in a spinal cord injury. There's a few publications out there in Japan, for instance, but the availability of primate models for looking at efficacy is just, it's very difficult. Looking at safety, for instance, tumorigenicity, it really wasn't also a practical consideration because we were wanting to look at hundreds of animals for teratoma formation and toxicity formation, and that would be limiting in terms of a non-human primate model.
Okay, last question, Michael.
So part of the story is just the process of bringing something into the clinic, and things, unanticipated things happen. And I was wondering if you could talk in a case study kind of way about how you dealt with the appearance of the microcysts, which obviously was not something that you anticipated.
Oh, yeah. This case study about the cysts, I can give you a little more, flesh that out. One of the things when we did some preclinical studies, we found that in some lots of OPC1 that we were producing, that we saw these cysts, and some at higher frequencies than others. And that's why I emphasized in my particular talk the characterization of cells and lots of materials, because we had retains of all of the lots, and we knew what their outcomes were in animal models, we were able to go back to that data, do deep dives into various microarrays and various RT-PCR array analysis to see what candidate markers were associated with the formation of those particular cysts. And we found them, and the reason why we're able to find them was because, we had representative samples from each of the different lots that gave cysts and didn't give cysts. And we were able to retrospectively pick out a candidate marker and then subsequently show in a prospective study that it was predictive, okay? And that's what kept us on hold for a couple of times. But I must admit it was a very good exercise and a very good learning curve to say, keep material that you have from all of your studies. You will use it in the future.
Okay. Jane, thank you for updating us and sharing with us about this important trial. Thank you very much.
Thank you.